Until the start of 2018, it is believed that DNA has all the information on everything. DNA sequencing technology is helping researchers in finding out the hidden information. By mapping out the whole genome of an organism, we now have a better idea of why giraffe has this long neck and why snakes are so long. Genome sequencing technology allows us to compare and contrast the DNA of different organisms and understand how they evolved and got such unique features. The same thinking applies to humans, with different skin colors, eye color, hair, height, and other variations caused by genetic material. But then we come up with a biological dark mystery, the dark DNA. Close to the dark matter, which covers about 25% of the universe and yet so far remains a mystery. And now biology has the same dark mysteries which are still unknown. Much of the genomic dark matter is thought to originate from ancient transposable elements and other low complexity repetitive elements. Uncategorized genetic material is found in humans and many other species. Their phylogenetic novelty could indicate the cellular organisms or viruses from which they have evolved. A puzzle posed by segments of dark matter in genomes, long winding strands of DNA with no obvious functions have teased scientists for more than a decade now. But in some cases, we are faced with a mystery. Some animal genomes seem to be missing certain genes, ones that appear in other similar species and must be present in the body to keep the animal alive. These apparently missing genes have been dubbed the dark DNA, and its existence could change the way we think about evolution. The fat sand rat is a strange creature. It lives in the burrows, eats about 80% of its own body mass each day, and doesn't drink water. But the really odd thing about this gerbil is that some of its DNA appears to be missing. In the recent study published in the journal PNAS, scientists looked at the one such case involving sand rats, a species of gerbil that lives in deserts of North Africa and South Middle East. Scientists looking for a particular gene called PDX1 related to the production of insulin to understand why this animal is particularly susceptible to type 2 diabetes. But when researchers look for a gene PDX1 responsible for secretion of insulin, they found out it was missing with 78 other surrounding genes. Scientists were stunned when they find these creatures are living healthily without these genes including PDX1, which are supposed to be essential for their survival. So the question is, where are they? They found out the genes weren't really missing but hidden somewhere in the genome because they found chemical instructions in desert systems which could only be produced by PDX1. The DNA sequence of these genes are rich in G and C molecules, which are 2 out of 4 bases, ATGC, which makes up the DNA. We know that GC-rich content are problematic for some DNA sequence technologies, which makes sense that these genes are not detectable rather than completely missing. Because of that, researchers call this region a missing part of DNA as a dark DNA. Digging deeper into the mysterious genome of sand rat, the researchers find out the unexpected number of mutations far more than other closely related species. GC content is much higher in mutated region, making it hard to detect for standard methods. Excessive mutation can often stop the gene from its working. The sand rat's gene somehow managed to fulfill this gap despite the radical change in the DNA. The dark DNA is rare but not new. It was previously discovered in the bird species DNA. In a study published in Genome Biology, researchers find out 274 missing genes in many bird species, yet necessary for almost all vertebrates. Once again, GC-rich DNA was detected, products of which are found in some body tissues even though genes are apparently missing. The conundrum has centered on DNA sequences that do not encode proteins and yet remain identical across all broad range of animals. By deleting some of these ultra-conserved elements, researchers have found that these sequences guide brain development by fine-tuning the expression of protein-coding genes. Dark DNA consists of dark proton sequences for which states of single DNA proton corresponds to those of DNA, mRNA, amino acids, and tRNA. Dark DNA is one of those speculative ideas of TGD-inspired quantum biology. Ordinary biomolecules would only make their dark counterparts visible. Dark biomolecules would serve as a template around which ordinary biomolecules such as DNA strands are formed in TGD universe. All basic biomolecules of genetics would be pairs of ordinary biomolecules and its dark proton analog. Although ordinary DNA is not out of ordinary genes, the dark DNA would still exist. If dark DNA actually serves as a template for the transcription of mRNA, everything is still okay after the knockout. Could it be that way? We do not understand even transcription correctly. Could it be actually occur at the level of dark DNA and mRNA? Dark mRNA would attach to dark DNA, after which ordinary mRNA would attach to dark mRNA. One step more, damaged DNA could still do the job. DNA transcription would have very little to do with biochemistry. If this view about DNA transcription is correct, it would suggest a totally new manner to fix DNA damages. These damages could actually at the level of dark DNA, and challenge of dark genetic engineering would be to modify the dark DNA to achieve proper functioning. Also, a translation could be based on the pairing of dark mRNA and dark tRNA. This suggests a fresh perspective to some strange and even ugly-looking features of the genetic code. Are DNA and mRNA always paired with their dark variants? Do also amino acids and anticodons of tRNA pair in this manner with the dark variants? Could the pairing of the dark matter level be universal and determined by the pairing of dark amino acids with the anticodons of dark RNA? Could the anomalies of the code be reduced to the non-uniqueness of pairing of dark and ordinary variants of basic biomolecules? No doubt you have heard of dark matter, which is thought to make up over a quarter of the universe. We know it's there, we just haven't been able to detect it. Well, something similar is afoot in the genome. Most textbook definitions of evolution state that it occurs in two stages: mutation followed by natural selection. DNA mutation is a common and continuous process and occurs completely at random. Natural selection then acts to determine whether mutations are kept and passed or not, usually depending on whether they result in higher productive success. In short, mutation creates the variation in organism's DNA. Natural selection decides whether it stays or not, if it goes, and so bases the direction of evolution. But hotspots of high mutation within a genome means genes in certain location have a higher chance of mutating than others. This means that such hotspots could be an underappreciated mechanism that could also bias the direction of evolution, meaning natural selection may not be the only sole driving force. The discovery of dark DNA is so recent that we are still trying to work out how widespread it is and whether it benefits those species that possess it. However, its very existence raises some fundamental questions about genetics and evolution. We may need to look again at how adaptation occurs at molecular level. Controversially, dark DNA might be a driving force of evolution. So far, dark DNA seems to be present in two very diverse and distinct types of animals, but it's still not clear how widespread it could be. Could all animal genomes contain dark DNA, and if not, what makes gerbils and birds so unique? The most exciting puzzle to solve will be working out what effect dark DNA has on animal evolution. So that's it for today's podcast. See you on the next episode.